I wanted to know if there was a general explanation. So we're all probably familiar with things like this. This is sort of current ocean and future ocean, right? So there's all these different changes. And we tend to think of all environmental change as bad, but if you're photosynthetic and you don't have to bother calcifying, actually things like more nutrients and more CO2 are likely to be very good. So for some subset of things in the environmental changes that we're going through, well, not pleasant for humans that rely on being able to predict what's going to happen to our ecosystem services, can be actually, for many of the organisms that live there who don't care about us, some of these environments are really improved. So let's talk about enrichment, right? We tend to do evolution experiments, and we tend to do most of our physiology experiments with this view that environments are always getting worse. We talk about starvation and deterioration. But I thought I would talk about enrichment, and this is partly because I enjoy enrichment personally, and partly because I'm a bit of a shit disturber. So evolutionary biologists, and as far as I can tell, ecologists, are obsessed with environmental deterioration. And I blame Van Balen, not Van Halen, but Van Balen, because he published something with this title, An Evolutionary Law. Who does that, right? That's just asking to be proven wrong. And an evolutionary law says that the effective environment of any group of organisms deteriorates at a constant rate, which is like, so environments are always getting worse. Right, good. Let's just take that and run with it. And we did. We ran with it for a solid 50 years. And all evolution experiments are done like this. You take a population and you plunge it suddenly. This is a fitness landscape, and basically high elevation is high fitness, low elevation is low fitness, and then on the other axes you would have traits, which can be either phenotypic traits or gene sequences or whatever you want, until you have so many dimensions you can't visualize a cube anymore. So all of the organisms I've worked with have 2 traits. But all evolution experiments take organisms or populations, they plunge them suddenly into environments which they are very poorly adapted to. This is sort of like taking penguins and moving them suddenly to the equator. And when they wake up the next morning, shocked to find themselves without any icebergs or fish, you ask what they do, because that's completely realistic, right? So our understanding of evolution looks like this. It's an increase in fitness. It's an exponential increase in fitness that we can predict and which never ends. It asymptotically is going to approach something it never gets to. So this is interesting, and it's super useful for understanding things like the evolution of antibiotic resistance, super useful for understanding things like heavy metal tolerance, maybe not so useful for understanding what happens when you start at the top of this peak and then somebody just moves the peak up. Right? So why would I study improved environments besides the sort of obvious chocolate payoff? One, if we're interested in future ecosystems, the organisms that are sticking around are the ones that are going to be winning. They're the ones that are actually going to see the environment as a good environment. So if you want to know what's going to be around, you want to study the organisms that are going to win. This is, like, to me, this is actually pretty intuitive, right? So this is an interesting paper by Steph Dukevitz, where basically they looked at the growth rate response to CO2 enrichment of these major groups of phytoplankton. I put pictures up: coccolithophores, diatoms, other large, which I'm assuming is this monster, diazotrophs, and then Sinecococcus and Prochlorococcus, the green dots. And what you see is that Sinecococcus, this green dot here, this blue bar, responds positively to high CO2, and so we get what I call the world of Sinecococcus scenario, where as time marches boldly forward and CO2 in the atmosphere goes up, the fraction of Sinecococcus in the ocean increases because it reacts to high CO2 by increasing its growth rate. So I, for one, feel like either we have to test this, and if necessary, prepare to welcome our new overlords. The other reason is that a lot of technological applications, so practical applications working with chlorophytes, often are focused on scenarios where you want fast growth and high biomass. So you're providing organisms with a vastly improved environment, and then you're cantilevering all sorts of commercial applications on that phenotype being stable. So we do really, it behooves us to understand when phenotypes are stable and when they're not. So let's try to get at this generally. Short-term winners. So Sinecococcus in the world of Sinecococcus scenario, right? Short-term winners. By definition, they're the ones that have a positive growth response to environmental change. We can talk about how realistic that is, ecologically incompetitive abilities and whatever, but right now this is what the models are using, so we're going to go with that. To have a positive growth response, you probably have to increase your metabolic rate. Is everyone happy with that? Yeah. If you increase your metabolic rate, you probably have to deal with more damage, more intracellular damage, which could build up gradually if you can't step up repair. And we've seen this before. I love putting this picture up and watching people go. Don't worry, we're not going to walk through these equations. What I'm going to get at is we've seen this before. This is the argument for why asymmetric cell division exists. The idea is that if you want to divide faster, you damage cell components more as you divide, and then to increase cell division rates, you make it garbage, which is that you put the used parts in the crap parts in one daughter cell, the good parts in another daughter cell, so the average growth rate of the population can increase in that way. So that's one solution, and that's mitigating damage. We've also seen it before in the evolution of germline and soma, so this macroevolutionary transition where all cells are able to reproduce to a division of labor where you have somatic cells that can't reproduce and germline cells that can reproduce. This is called the dirty work hypothesis. And what I'm showing you here is a simulation done at Ben Kerr's lab by a really awesome graduate student named Heather Golby. And the circles represent cell lineages that are propagule eligible, which means that they can divide. And the color is the amount of metabolic work those cells are doing. And what you can see is that as soon as cells are willing to become propagule ineligible, so they become squares, which means that cell can no longer reproduce, but it can give all of its work to a cell that can reproduce, is suddenly the amount of metabolic work that can be done increases dramatically. And then what's going to happen is most cells become unable to reproduce. So that's the evolution of a germline. So that again is the idea of if you want to increase your metabolic rate, if you want to increase your capacity to do work along that grow faster, you need to deal with this damage somehow.